Thank you, Bill, very much for this very kind introduction. And good morning, everyone. It's a real pleasure to be here. It's been about 10 years since I've been to Seattle, so it's really nice to be here. So today I'm going to talk to you about some of our work in multimodal neuroimaging of brain anatomy and function in awake marmosets. And I don't know why the slides are not advancing. Oh. But that's the other one. Okay. All right, so the outline of my talk is I'm going to show to you that the marmoset is an animal model of functional brain activation using MRI. It can be imaged in conscious awake conditions, very much like we study humans using MRI. And we're going to talk about some anatomical work we've been doing that has shown remarkable cortical architecture using T1-weighted MRI. And then go into the functional MRI side of things to study the brain and any of the sensory systems that activate so well with the marmosets. And then we're going to merge into more detail of the neurovascular unit and look at some direct visualization of blood vessels and neurons using two-photon microscopy. And that will set up the need for the third part of the talk, which is the development of transgenic marmosets that express dysfunctional calcium indicators that have been so important in telling us what neurons are doing in terms of functional brain activation. So one of the main motivations for studying primates is that they're phylogenetically closer to humans, and the marmosets do retain a lot of these advantages in terms of its anatomical and functional organization. The marmosets have the primate-like parcellation of the brain, and this is what this slide alludes to, where you can see that you can parcellate the marmoset brain and find homologous regions to the ones that you find in the more commonly studied primate, the macaque, and all of these relate to the organization of the human brain. So working on the animal model side of things to try to understand human brain function and organization is the main goal. But the marmoset as an experimental model also resides in a very important and privileged site, because it's situated right in between in terms of neurocircuitry complexity, simpler animals such as the zebrafish and mouse, where the development of genetic techniques has been so well established that you can learn everything at the genetic point of view from these animals. But as you go up in this chain of neurocircuitry complexity, you lose this power of doing genetic manipulation and trying to understand functions of specific genes. And fortunately for us, now these techniques are becoming amenable to being used in marmosets. And this comes from a seminal work by the group of Erika Sasaki and Hideyuki Okano in Japan, who demonstrated in 2009 that it's possible to generate transgenic marmosets. In this case, this was a proof of concept that showed a GFP expression throughout the entire body of the animal. But the real main result of this work is that the transgene expression actually passed on through the germline. So now there are lines of transgenic primates that are being generated. And these really boost the power of the use of these animals in neuroscience research. Another main advantage for us who are interested in understanding and studying the brain is that the marmosets are lysencephalic. So of all the non-human primates, these are the animals with the lowest degree of cortical folding and cortical gyration. And because of this, it makes the localization of brain function using fMRI or optical imaging or even electrophysiology trivial, because everything is there on the surface and you don't need to spend a ton of time trying to properly localize your electrodes to the right place and deal with models of inflation of the brain to get rid of the gyration. And well, let's get started on how do we do this in the marmosets. Basically, when the animals go in the MRI, there are two main types of setup that we use. They can go anesthetized, and these are very typical studies for anatomy, where we use isoflurane as the main anesthesia. It's very safe to be used, and the animals recover from it very quickly. But one of the problems with isoflurane is that it suppresses neuroactivity to the extent that it's very hard to study brain function when the animals are with isoflurane. And if that's the goal of using in anesthetized animals, there are other anesthetic regimens that can be used, for example, propofol plus fentanyl. But as I mentioned in the beginning, one of the main goals is to basically study these non-human primates the way we study humans. And for this, we felt it was going to be very important for us to develop protocols in which we could acclimate the animal to going the magnet awake. And this is now the bulk of our work, where we can do both anatomical or functional studies with unconscious animals. In either case, we pay a lot of attention to the physiology of the animals so that we make sure that they are as comfortable as possible in there. And that means monitoring temperature, heart rate, pulse oximetry, and tidal CO2. Make sure that the data is acquired under relevant conditions. For having the animals in the magnet in the conscious awake condition, we have developed a MR-compatible cradle that consists of a body cover where we dress the animals in a sleeveless jacket and zip tie them to this body cover so that they don't move backwards or forward. And then to restrain the head so that we can have decent quality images acquired, then we took advantage of the 3D printing for custom designing helmets that basically are composed of two parts. The red part here supports the head of the animal, while the green part supports the chin. The animal goes in a magnet in the Sphinx position, which for marmosets is a very natural position of being. And with 3 weeks of acclimation, they are ready to be studied with MRI. This is a picture of one of the animals ready to go in the magnet, where you see here the two-part helmet with the chin piece supporting the chin of the animal in the Sphinx position. We got here electrodes for doing somatosensory stimulation. And the top of the helmet here, we line it up with foam for added comfort and also for some suppression of the MR-induced noises. And once we put the animal in the magnets, this is what they hear. And we, in addition, put some RF detectors on top of the helmet here so that we can collect the signals coming from the brain. And as you can see, the animal is doing exactly what humans do when they go in the magnet. They doze off because it's so boring in there, right? So much for awake imaging, but you know, at least we know that the data is being acquired under relevant conditions and without the confounds introduced by anesthesia. I should also mention that in addition to the effects of anesthesia and neuroactivity, you have effects of anesthesia in the way that the blood vessels respond, and that is another confound as well. So the system has worked really well, and the cradle became more and more elaborate. It's like, you know, when you first buy a car, you buy the simplest one that you can afford, but then later you start introducing all the gadgets in there. So these days, the helmets became more elaborate because we put the RF detectors on the inner surface of the helmet and not on the outer surface, so that we have more sensitivity from our MR images. And we've introduced also the ability for the animals to behave inside the magnet. So for example, this is a setup where the animal is undergoing a visual stimulation. There is an LCD monitor outside the magnet, and they have a clear path to see what's on the screen. And we have an IR light source and an eye tracking camera so that we know that the animals are paying attention to the stimulus. And the data that we get from this kind of setups are really outstanding. This is an example of fMRI of the somatosensory pathway in a marmoset with an in-plane resolution of 250 microns per image element here with a slice thickness of 1 millimeter. So we see all the regions that activate well with somatosensory activation, such as primary somatosensory cortex, secondary somatosensory cortex, and also caudate activating well, in addition to thalamus, which we don't show here. And all of this is afforded by the extra sensitivity that we have in embedding these detectors on the inner surface of the helmet so that they are in closest proximity to the brain, which is the area of interest. So as you can see here in these SNR maps, we have maximum sensitivity right in the cortex where the activation is all happening. And this is all work that is developed in-house. And this also boosts the sensitivity for anatomical MRI of the brain. So if you go to the clinics and you have a neuro MRI done, one of the techniques that they're going to use is so-called a T1 weighted image that has different acronyms depending on which vendor supplies the sequence. But basically, this is a sequence in which the rate constant T1 is used to differentiate gray matter from white matter from cerebrospinal fluid. And as you can see, you have excellent gray to white matter contrast in these images. So when applied to the marmoset, we can acquire these images in 3D mode of the whole head with 150 microns isotropic resolution in 51 minutes. So with this, you can have this map for each of the animals and compare, for example, with diffusion tensor imaging that is acquired ex vivo in 36 hours with the same resolution. You see here the main white matter tracts and the directionality that is used in tactography algorithms to infer connectivity between different regions of the brain. But again, when you resort to the in vivo image, you see excellent contrast again between gray matter, white matter, and CSF. I would like to also point your view, if you can, now to only gray matter, and you'll see that gray matter doesn't appear uniform in these images. There is a lot of contrast within gray matter in itself, and this is due to the fact that the cortex is highly myelinated. All these white matter fibers that are making these long-range projections between the different areas of the brain, they actually arrive into the cortex in myelinated form. And using this technique, then we can acquire this image in 3D mode. And if we compare the contrast with post-mortem histology for myelin, you see that all the areas that are heavily myelinated in the histology, indicated here in dark staining, appear bright in the MRI. And there's a lot of myelination, as I said, in the cortex, so areas such as the frontal eye fields and primary somatosensory cortex, primary auditory cortex, area MT, and area DM in the visual extrastriate cortex, and the stria of Gennari in primary visual cortex are heavily myelinated cortical areas that show up really well in the in vivo MRI. And because these images are acquired in 3D mode, then we can do a surface intensity projection of these images and show this in terms of 3D surfaces. And what you see here with the color coding is that the areas that are brighter are the areas that are mostly myelinated. And what you see then is what we call this Cortical Myeloarchitecture Map that basically separates the different functional areas of the brain and of the cortex very well. And in fact, myelin is one of the oldest stains used for parcellating the brain. It started with the pioneer work of Francesco Gennari in the late 1700s. And since then, it's been used as one of the gold standards for parcellating the brain in terms of its anatomical organizations. So what we see is that this anatomy is highly related to the functions because we can see the separation between the primary functional areas. For example, if we look here now at this a digitally flattened view of the brain, we see very well that V1 is heavily myelinated, and because V2, the secondary visual cortex, is not that heavily myelinated, we see a very well-defined border between V1 and V2. And you see area MT and area DM associated with the visual pathway for processing of different features of the images that the animals see. And from the foveal area V1, you can even, if you squint your eye, see a tract that communicates to MT and another tract that communicates to DM. And as you go towards the front of the brain, you have the primary somatosensory cortex, S1, with three main areas of myelination separated by septums that are not so well myelinated. It is thought that these areas of myelination correspond to the foot, the hand, and the face representation of the somatotopic body representation. You have a primary auditory cortex, area A1, and also area R here. You have the secondary somatosensory cortex inside the temporal sulcus, and you even have the motor cortex, M1, in there represented well. So with these maps that I said takes 51 minutes to get, you have now laid out the anatomical definition of the primary sensory areas, and you can use that to do a number of different things. For example, you can do parcellation of the brain and quantify these areas. And what we've observed is that in the marmosets, about 25% of the cortex is dedicated to the processing of visual information. If you add areas V1 and MT and DM, you have about 25% of the brain. So vision is very, very important for the marmosets. But also the auditory cortex, the marmosets are highly vocal animals, and vocal communication is very important for them, especially for keeping the group together. They are arboreal animals that jump from tree to tree, and they don't have direct visual contact with all the members of the group, and therefore this vocalization is very important in terms of keeping the group together and of warning the different animals, both for the presence of predators and also for the opportunity of food resources. So these areas being heavily myelinated, presumably the myelin is there to speed up the conduction of the signals even within the cortex, is something that is functionally very, very important. And then how do you relate this anatomy to brain function? So one of the first experiments we did was to try to understand how, if we stimulated the animal and in an fMRI experiment, how the areas of fMRI activation would relate to the anatomy that we see with the myelin mapping. So just to remind you, the primary somatosensory cortex has these three islands of myelination. So the most medial one near the midline is the foot representation. Then you have the hand representation more laterally located, and even more laterally the face representation here. So if we use fMRI and we stimulate the leg of the animal, then these are the areas that show up right here in this medial representation, as you can see. And then if you now stimulate the hand or the forearm, the fMRI shifts to this more laterally located here. So you have the T1 anatomy guiding where to look for in the fMRI signals. And this, if you look now at the representation of the hand here in this 3D surface, you know, the activation will show right where you expect it to be. As Bill alluded to, we have a very, very strong interest in understanding the nature of fMRI signals, and that has helped us develop the new techniques that can be used to study any brain. And we have a very, very strong interest in understanding the biophysical mechanisms that are behind the generation of these functional signals that can be picked up with MRI. So we do a lot of this work in the somatosensory cortex because of the maximum sensitivity that we have, as I mentioned. And we've been looking a lot at the temporal and spatial evolution of the signals. So this is an experiment where we have a very short somatosensory stimulus, and we look at the bold response both in the space here and in time to understand, how the signals are evolving. And with this, we can pull out this, what we call the hemodynamic response function, HRF, and we do a full characterization in space and in time. We know exactly how does it start, what is the onset delay, and we know how long it takes for it to peak. We know how wide it is for the stimulus duration, and we know from which layers of the cortex the signals are coming from. And this is very important for us to understand this biophysics of fMRI activation. So, for example, one of the works is relating this fMRI signals with respect to the laminar profile of the cortex. And what we see is that the hemodynamic response signals have different amplitudes and different onset times depending on which layer of the cortex you are. We also understand how the signal evolves. So this is a cartoon that shows the areas of activation as a function of the stimulus duration. So here the numbers are the number of electrical pulses that we do at a frequency of 64 hertz. So 64 pulses here would be a 1-second stimulus, which is extremely short to what is done typically in human fMRI experiments. But that allows us to basically understand how this fMRI activation area, this bold activation area, grows with the stimulus duration. And what we see is that by about 1 second, the stimulus are pretty much saturated in terms of its spatial components, and increasing the number of pulses to 2 seconds or 4 seconds basically doesn't add much in terms of the total area of activation. And we can see here in this plot that shows the number of activation voxels as a function of the stimulus duration, that you have this activation for stimulus that are as short as 1 second. And this is the regimen that we like to work on, which is a regimen that, in our understanding, is minimum perturbation of the system, so that we understand signals that are really relevant to how the brain works, instead of using signals that basically are dispersed over very long stimulus durations, and that could have confounds in the interpretation of the results. In addition to somatosensory cortex, we can also do auditory MRI in a marmoset. So this is work that we've been doing in collaboration with Corey Miller at UCSD and David Leopold and NIMH in Bethesda. And these maps here shows activation in the primary auditory cortex of the marmoset. And what you see is 2 clusters of pixels, one in blue and the other in orange. That shows the areas of the primary auditory cortex that respond either to low frequency tones or high frequency tones. So you have this alternation of high to low to high that is very typical of the organization of A1, and we can pull that out, in the marmoset, even though MRI is thought to not be the best way to do auditory studies, because MRI in itself is a very acoustically loud technique, but you see that you have very high quality data that can be obtained from the marmoset. And then we can also study functional connectivity in them as well. So this is an example of work that we did in collaboration with Elliot Stein at NIDA, where we were interested in probing these resting state networks. Basically, you're collecting a time series of data and just later asking which areas of the brain are sort of oscillating together with more or less the same frequency of the same phase. And if you do this kind of question and you do analysis using independent component analysis, principal component analysis, or some VBM algorithms, as well as has been mentioned in previous talks, here we can pull out these resting state networks. And the marmoset has at least 12 of them that make anatomical sense and that correlate really well with RSNs that are obtained both in the macaque and the human. So there are lots of visual areas. There is a basal ganglia area here, somatomotor area. There is the default mode network, which is an area that is thought to always be on under normal conditions in the brain. There is a cerebellar network. So, in summary, really, you see this is data that further confirms that the functional organization of the marmoset is very much like that of the human, and these animals are perfectly valid animals to study in neuroscience. And then in visual stimulation, has been a more rewarding work for us, just because the animals are now behaving in a magnet. When you present a somatosensory stimulus or on an auditory stimulus, you don't really ask whether the animal got this stimulus or not, because the activation of the brain shows that they got it. But in the visual stimulation, you have to first ensure that the animal is paying attention to the task and is not closing its eyes. So we had to introduce a positive reward system here, and that basically is the delivery of sugary fluids. Marmosets, they love marshmallow, they love anything that has sugar. So if you just basically reward the animal by delivering sugar during the presentation of the images, and if the animal closes the eyes, then there is no delivery. This positive reward works wonderful to ensure that the animal is paying attention to the center and to the screen, and I'll show this in the next slide. But basically, one of the very first experiments that we did was to try to understand whether different areas of the marmoset visual cortex were taking care of parcellating the scenes based on the structure of the images. So if we present, for example, images of other marmosets, what areas of the visual cortex are responsible for processing information of faces versus body parts versus familiar objects. And these are our controls. And we did this study in parallel in two cohorts of animals. And in one of the cohorts of animals, we used electrocortical encephalography to look at the data, ECOG data. So the stimulus paradigm was a block design where you had these interspaced periods of rest and stimulus, and each stimulus block was 16 seconds. I think I'm losing the pointer. I wonder if there is a better pointer for me. Within each 16-second block, every 3 images, the images were changed randomly every 500 milliseconds. If the animal was paying attention to the screen, then there would be a reward in the form of a drop of juice. And here's how the animal behaves in the magnet. So you have the liquid reward system, and you see that during the rest, there's nothing, so the animal is just scanning, and if nothing happens, the animal gets bored, like any human subject does. But then once the fixation point comes, you see how well the animal focuses, gazes, it's in the center of the screen, and then the stimulus comes, and then the reward comes, and then the animal takes the reward. Thank you. And in doing so, so we guarantee that the animal is paying attention, so the behavior is right there. And then once the stimulus block is over, then you're back here, and the animal gets bored again. And also a typical day at work for them. And what kind of data do we get? So it's really tremendous, and we're still parcellating all of it, but you basically get activation both in the primary cortical areas and also some subcortical areas, such as the thalamus and the pulvinar areas. The LGN areas activate really well, and then here you have activation through all the main visual areas: primary V1, V2, V3, V4, MT here. All the areas in the extrastriate pathway are responding well. One of the interesting features that we saw is that there is some sort of hierarchy of these different areas, so that the areas that are more occipital to the cortex here around V1, they don't really care what the image is. They are always activated, and therefore we built an index called the structure selectivity index. And the lower this number is because anything activates them, but the higher this number is because these areas become really selective to what they see. So as you go from the occipital areas of the brain towards the more anterior areas, the areas become more and more selective to the point that the most anterior areas only care about a certain category of stimuli, for example, faces. And if we ask then what areas have a higher fMRI response for faces versus the different categories, then we get this discrete set of patches, and we found five of them with fMRI, and one more with ECOG that was in an area that wasn't favorable to the fMRI because of signal dropouts. But these areas here, they have higher percent signal changes that you measure with fMRI when faces are displayed versus when anything else is displayed, like body parts or objects. And as you go from the most occipital area, area O, to the most anterior area O, then you see that these areas become really selective to the point that the most anterior areas pretty much only care about faces, and they don't care about anything else that is shown to the animal. And very quickly, just to go through the ECOG data, with ECOG, you have this electrode arrays that you surgically implant underneath dura, and they just sit on the surface of the cortex, and these electrodes, they can work for years, so you can use the same subject in a number of experiments throughout the years. You collect these evoked field potentials whenever you present a visual stimulus, and then once you get this stimulus, these responses, you can do a frequency analysis. So basically, you look for the amplitude of these evoked response potentials at different frequencies if you do a Fourier transform in one of the directions. And what we see is, again, like in the fMRI, there are sites in the ECOG array that show preference for faces versus body parts versus objects versus scrambled control images. And you see that for different stimulus durations. And then if you look at the amplitude of these signals as a function of time, you see that indeed in this site here, when you present faces, you have higher amplitude than when you present objects or body parts. So then you can do a map of each of these ECOG electrode sites. And here we have in yellow and blue data from two different animals, Marmoset F and Marmoset S. And then you can look at some sites, for example, site 1, which has somewhat of a preference for body parts versus faces versus objects, while if you go to site 6 or site 8, site 6, you have higher preference of faces. And you can do this color coding, and what we have here, according to the wheel, is that you have the higher degree of selectivity again when you go from the occipital areas to the most anterior areas. But once you get here in the most anterior areas, you have really this cluster of sites that respond mainly to faces, while there are other cluster of sites that respond either to body or to objects in between them. And if you compare the clusters that you obtain with ECOG versus the cluster that you obtain with fMRI, they're very analogous. They correspond really, really well. So it shows how good a quality of data you can obtain from these animals. Now, let's move away from fMRI. fMRI is a great technique for looking at this aerial functional representations of the brain, but we know that the underlying signals are, individual blood vessels communicating with individual neurons or vice versa. And for this, we need to get this kind of data, we need to resort to microscopy, and we've seen a few talks already on using two-photon microscopy to directly visualize the neurovascular unit. So this is data obtained from a mouse where you see the entire vasculature. This is over somatosensory cortex in a mouse. This is a view from the top. This is a view from the side, where we can go up to about 700 microns or so. The whole field of view is 600 microns, but you have you have actually an isotropic resolution that is better than 1 micron. So you can really see not only the large vessels, but you can see the capillary network really well in these animals. And then one of the greatest tools that has been developed is a way for us to not only visualize the neurons, but visualize neuronal function, and this is with the introduction of this genetically encoded calcium indicators of the family GCAMP. And we also seen a few talks already in this conference about the use of GCAMP to study individual neurons. So here in our experiment, we have an intravascular fluorescent tracer that we usually use Texas red dextrans. They show up in red, showing us what the vessels, where the vessels are and what they're doing. And GCampin 6 is marking the neurons. And when you do this in the mouse, you have this time movies where you see the activation of the neurons, and simultaneously to the activation of the neurons, you can collect data on the blood vessels, and then later you can interrogate, when this neuron activates, for example, which vessels respond and how do they relate to each other. So we're porting these experiments to the Marmoset. The preparation is very much similar. You just implant a cranial window in the animal. Unfortunately, just the helmet now is not sufficient to hold the animal still enough, because if you want to collect data with better than a micron spatial resolution, you've got to really make sure that nothing moves, and therefore we need to use head posts for that. But this is an animal there, and as you can see in the movie, the animal's awake, it's looking at different places here. Here's the cranial window, and the animal is absolutely relaxed because, nothing is happening there, but also because, there is nothing to happen. This is, from the point of view of the imaging acquisition, also very non-invasive. And the data that we obtain from the Marmoset is not as good as from the mouse, and this is because there is more scattering. There is also more motion with the animal being awake. But fortunately for us, this motion is highly restricted to the very top surface of the brain where the large vessels are. As you dive into the capillary network, and you see here now the capillary is coming, the motion decreases a lot. But still a problem that needs to be corrected post-acquisition, and therefore, this is one of the main challenges of this big data kind of data processing algorithms that we need to do. So in the Marmoset, to inject the GCamp6, to deliver the GCamp6 to the neurons, thus far we have been using these AAV viral constructs, and basically we do a stereotaxic injection over the area of interest, for example, the hand representation of the somatosensory cortex. And this is the area of the cortex that basically you target with the GCamp6. And if you look post-mortem, you can see that the virus made this GCamp6 be well expressed. This is colocalization with NUN. NUN marks the nucleus of the cells, and here we have the cytoplasm and with GCamp. And we can then look at a larger area of the brain by tiling these acquisitions serially here. And what is really important is that this preparation is stable over a large number of months. So this is showing that over the somatosensory cortex, we obtain more than 6 months of stability of the preparation. We see here the optical clarity of this window is maximum in days 2 or 3 post-surgery, and it degrades with time, so that by 6 months, most of our optical clarity has gone away, and this is mainly because dura regrows, and we lose the clarity. But this is the data that we obtain from the Marmosets. Again, the neurons are marked with the GCamp6, and the vessels in red with the intravascular Texas red dextrans. And we can see here the red blood cells flowing through the capillaries. Again, the problem of motion, that will be great to take advantage of some of these new algorithms being developed for doing this kind of data analysis. But basically here, you see one of the cells that is filled with GCamp. And when we stimulate the animal somatosensory stimulation, we see an increase in the fluorescence coming from that neuron. And then if we look at the blood vessels, a single vessel coming right here, when the stimulus is on, you see that the vessel dilates. So again, relating the neuroactivity to the vascular dilation is one of our major goals, and we've been very busy analyzing this kind of data nowadays. But one other data that we can obtain is looking at the entire microvasculature of the marmoset, and we pull a lot of data out of it. For the average capillary length, for example, it's about 80 microns with a capillary density near 7,000 capillaries per millimeter cubed. And we can even ask in here, you have these arterioles represented in red and the venules in blue. we can ask for each penetrating arteriole, what is the range of capillaries that are fed by that arteriole? And you see that this microvasculature is parcellated also in functional microdomains, and it will be very interesting to understand how these functional microdomains on the vascular side relate to the functional architecture of the cortex, and what happens, for example, if that feeding arteriole was obstructed, for example, in stroke, what would happen to blood flow in this capillary domain? So it's one of the things, one of the areas that we're going through as well. But now, as I mentioned, to do this in the marmosets, thus far, we've been using viral approaches to deliver GCAMP6 to the brain. And that has a number of problems. One of the problems is that there is a tropism of the virus that makes the expression of GCAMP6 to be not uniform throughout the cortex. Another problem is that we can't control how much is expressed, and one could argue that the viruses could make the expression so overwhelming that you interfere with the normal physiology of the experiment. So for this, then, to avoid this problem, circumvent these problems, we developed transgenic animals using constructs that we could apply to the marmoset. So this is an example of a mouse that expresses GCAMP6 under the control of the synapsin promoter. And you don't see much green in the cortex at all because the levels of expression are much diminished with respect to other areas, such as the olfactory bulb here or the hypothalamus here. But presumably, these levels of expression would be more physiologically relevant, not interfere with the normal physiology of the cells. So because of this, then we decided to try the transgenic approach and in a marmoset and make marmoset lines that express the GCAMP. And there is a whole cascade of events that needs to happen here. We need to be able to collect embryos from a wild type animal, do the viral injection in the embryos, transfer the animals to a foster mom or a recipient female, pray that this animal will get pregnant and will take the pregnancy to term so that babies are born, and then we do the analysis of the babies. And we got really good with this. I'm going to skim through these slides, but basically we can collect these embryos non-invasively. If you pair a male and a female, allow them to mate naturally because we know the cycle of the female, we know exactly when to collect this, and we got really good about collecting the expected number of embryos every time we attempt to do this. These are examples of these embryos that we collect and then do the viral injection at the embryonic level here. We can also do surgery and collect unfertilized oocytes that then we do in vitro fertilization and collect these embryos. With the advantage here is that not only we collect many more oocytes from every female, but also that after IVF, we have access to the embryo at the single cell stage so that we can do our introduction of the transgene right here, and we should have less mosaicism with this. So in doing a lot of these experiments, and out of 275 embryos that were transferred into recipient females, we got 8 live births. And these are 5 of these animals here. We have basically 3 lines. One line expresses a previous generation of GCAMP called GCAMP5. The other 2 lines express GCAMP6 under the control of the ubiquitous promoter CMV or under the control of the synapsin promoter here that should make the expression only in the neurons. And then we did an analysis of transgene integration, and basically every tissue that we see from the CMV expressing animals express the virus. And then we know also the integration sites, and there are multiple because this is random integration. And we also see integration of the transgene, for example, in peripheral cells, like whole blood peripheral cells as well. And doing post-mortem analysis of animals that either were stillborn or that died shortly after being born, we see also expression of GCAMP6 in different parts of the cell. So for example, in the spinal cord here, we see neurons with GCAMP expression here and UN in the nucleus. And one of the important aspects is not to do this for every animal that you do, but once you make one animal, that animal will pass this transgene on the germline. we were fortunate to be able to confirm this. So these were embryos that were collected from one of the founder, or from two of the founder animals that we obtained, and we saw that the embryos were also expressing the transgene, so there is germline transmission. These animals were then we followed up the pregnancy with ultrasound and saw, the presence of babies and in the uterus of the foster moms, and now these babies are born. We have 5 such babies born already on the F1 generation, so the lines were made, and we're really eager to utilize these animals soon in 2-photon microscopy experiments. On just one last slide on the functionality of the transgene, we haven't done that in vivo yet because the number of animals we've done with this is very restricted, but we've collected cells from the animals, and we analyzed the cells. We challenged them in vitro with ATP, an ATP challenge, and we see increasing fluorescence upon the ATP and exposure, and then once ATP is removed, the fluorescence quenches, showing that the transgene is functional in the cells derived from the transgenic animals. So I'm out of time, and this is my concluding slides. I hope that I showed to you that the marmoset is becoming a very important experimental animal model for basic science and translational research. The lysencephalic cortex, in my opinion, is a great advantage that facilitates the study of the neural circuits with any imaging techniques, MRI and optical imaging techniques. This germline transgenic marmosets expressing GCAMP have been generated, and we believe that they will be invaluable non-human primate models in neuroscience, allowing chronic in vivo monitoring of neuroactivity under conditions that are very relevant to the way that humans work. This work, while we started with GCAMP, we're expanding them. We have a very strong interest in neurological and psychiatric diseases, and they're coming up as well. And finally, I just want to thank the people that really did a lot of the work. Cecil Yan for the fMRI contributions here, and Jung-Eun Park for the work of the transgenic marmosets. I want to thank you again for listening to my talk, and I'm ready to take any questions. Thank you.
Yeah, there are a couple of microphones for making things.
That was a great talk, really exciting to see the transgenic marmosets. And I'm wondering, it seems like the combination of having windows over mapped sensory areas that are clear for so long, especially in an animal expressing GCAMP, it seems like the possibility to study learning and perception is really exciting. I wonder if you could comment on how the marmoset community is evolving in terms of training these animals to do behaviors, psychophysical behaviors, to connect behavior and the neural activity to take advantage of the approach.
Oh, thank you very much for this question. It's indeed a very relevant one because the marmoset community is a very open community and very inclusive of everybody. So there is a lot of work going on right now in 2 main domains. One is, or maybe I should say 3, on the sensory aspects, there is a lot of interest in understanding the meaning of the vocal communications. This work being led by Xiaoxin Wang at Hopkins University and also Corey Miller at UCSD, where they want really to understand how the animals communicate with each other and try to pull out things that really relate to how behavior from one animal interferes with behavior of another. One example of this is this cocktail party where you have 2 animals, one talking to each other, and then you bring a third animal into play and how this third animal interferes with the communication of the 2 others. The other example that I know is from David Leopold's work in visual cortex, where he's trying to understand how the animals look at naturalistic scenes. For example, if you play a movie to the animals, do areas of the brain always respond the same way every time a certain scene or a certain part of the movie is displayed, and how the number of exposure of the animal to the same movies change with time, the response of this neurons. And finally, there is also work by Angela Roberts at Cambridge University in UK, where she is looking at some aspects of fear conditioning in the animals and how that influences their behavior. But it's very rewarding indeed. And as you said, the ability of following up these animals for months, if not years, is a major, major plus.
Hi, I'm interested in the heterogeneity of myelination in gray matters you mentioned earlier in your talk.
Yeah.
I presume it's stereotyped across different animals in the same species. Is that correct?
Across animals of the same species,
Yeah.
there is some variability, but I tried to show
Yeah.
a table that showed that it's very well confined so that the areas do have sort of a common shape and size, and yes, there will be some animal-to-animal variation, but that is relatively small.
How about, are there data across different species? And also, what is the meaning? Does that reflect just axon density or activity or combination or other factors?
So as I said, myelination is one of the oldest stains that is used to parcellate the brain. And in the beginning, these brains were parcellated just anatomically. So for example, the Stria of Gennari and then Brodmann also used myelin to look at different areas of the brain and saw that there was a very strong conservation of the size of these areas so that he could come up with the Brodmann atlas, of the brain, based on this parcellation. I think you asked what is the purpose of this myelination, and we see that the heaviest areas of myelination are the sensory areas, and because of this, we think that myelin is really there to keep the speeding up of the transmission of signals. For example, you could think marmosets are arboreal animals, and they need excellent hand-eye coordination so that they can jump from one branch of the tree to the next without falling. They need to be able to quickly see a predator. They need to be able to also quickly see a food opportunity and use that. And I think the myelination speeds up the conduction of signals.
I'm wondering about the windows. You mentioned that after 6 months or so they start decaying in quality. I'm wondering whether does it affect the data and how it affects the data, and what sort of techniques can we do to try to keep the quality more stable over time?
Right. So in our hands and these experiments, the main reason for why we lost the optical quality was because dura, which we resect in the first surgery when we do the cranial window, starts regrowing. So if you could come up with a way that prevents that regrowth of dura, that would be great. Even better would be if we could come up with some sort of clarity-like technique that we could apply to the animal during the surgery so that would keep dura translucent so that we have penetration. One of the problems with two-photon microscopy is that depending on what wavelength you're using of light, and the G-camps are in the green, is not very favorable. You have a lot of scattering, so you lose signal and sensitivity very quickly. And in the mouse, some people report 300 to 500 microns. Some other report better. In a marmoset, with the G-camp, we can see to the bottom of layer 2, and then we lose signal with the G-camp. With the intravascular traces, which are in the infrared range, we can see deeper to 500 microns, but with the G-camp, we can only see to the bottom of layer 2. So it would be great to come up with techniques that prevent the regrowth of dura, or to even avoid resecting it right now, but right now we can't do it.
Okay. One more quick question, and then
I just wondered could you comment on the needs for noise canceling algorithms for the auditory stimulation, and whether you think you'd be able to look at that? They have a rich repertoire of vocalizations with different contexts, fee calls and alarm isolation calls, whether you'd be able to use the imaging to look at those networks of...
Yes. So on the second part of the question, yes, we have been playing different types of calls to the animals in the magnet and observing different areas, not only in primary auditory cortex, but also towards the prefrontal cortex that respond differently, whether you're playing a fee call versus a thrill versus another type of call, whether you're playing a call from a known marmoset, a marmoset known to that individual, or a completely new marmoset, so there is basically a lot of low-hanging fruits that are right there, and this work, we're starting to do it right now. And the first part of the talk was the noise cancellation. So the helmets that we use, they have this embedded foam that really takes at least 13 dB. It's not a lot, but it's quite enough. The animals have not shown to us any overt signs of being stressed out by the fMRI part of it, but we do use this MR compatible headphones that they are small enough that we can put right against their ears, and we have not had to use any noise cancellation techniques yet. It's possible that they would help, but thus far we found them not to be necessary.
Okay. Well, I'd like to thank the speaker again for a wonderful talk. Thank you, Afonso.
Thank you.